As we start talking about the different categories of biomolecules, we're going to start with the simplest ones first. That's the carbohydrates and the lipids. Carbohydrates and lipids are both excellent sources of energy for living things. They rarely have any elements other than carbon, hydrogen, and oxygen. You will occasionally see something else in one or the other, but not very often at all. Carbohydrates include sugars and starches. They are actually the main fuel that your body uses and that most living things use for fuel. The monomers of carbohydrates, of course, are monosaccharides, and the polymers are called polysaccharides. This involves things that we use as food, but then there are also some of the polysaccharides are very important for structure of various kinds of things in living in organisms of various kinds. And so it's important for us to understand both of those functions as they're very important. Monosaccharides are usually formulas of CH2O. Okay, if you think about it for a minute, that's where carbo and hydrate come from in the name carbohydrate, from the carbon in the water. The hydrogens are usually twice the number of oxygens, and the number of oxygens is usually pretty close to the number of carbons. And so when you see formulas like C6H12O6 or C12H22O11, you'll see that they're pretty close to that 1 to 2 to 1 ratio of carbons to hydrogens to oxygens. Monosaccharides are classified by the number of carbons they have. They're also classified by the location of the carbonyl group. Here you have two sugars, glucose and fructose, that have the same molecular formula, but their structural formulas are slightly different. The glucose has a carbonyl group on the terminal carbon, and the fructose has the carbonyl group on the second carbon. And so even though they have the same molecular formula, they are very different sugars. They have different functions because the functional groups make them different. That's what functional groups do. They often, at times, what you see is a linear structure here. This is glucose once again, okay? And that's the way we oftentimes show them. But when you put that sugar into an aqueous solution, which is where you usually find them, what happens is that linear structure forms a ring, like you see here, and so there's our glucose ring. So when you see a hexagon or a pentagon or a shape like that in a molecular, in a structural formula, you can generally realize that that's going to be a sugar unless it's got some other stuff stuck in there. So carbon, hydrogen, and oxygen. Notice that the oxygen here, once it forms the ring structure, the oxygen is in the ring, and that generally is what you will see with monosaccharides. And notice also that the carbons are numbered clockwise from that ring. So here's the oxygen, and then we have carbon number 1, 2, 3, 4, and 5, and then 6 is attached up here at the top. And that's generally not that necessarily that important to talk about, except you will need to know that when we talk about DNA structure and how DNA replicates itself and is transcribed and translated. That is important to understand then. So here's a ring structure. This is an abbreviated ring structure. The reason that some of these bonds look bigger than others is because it's to show that it's a three-dimensional molecule rather than in 2 dimensions like this one shows. That's just showing that you're seeing this closer to you and that part farther away. Disaccharides are formed when you join 2 monosaccharides together, and guess what? The type of reaction we use to do that is our old friend, the dehydration synthesis. Okay, we're going to take a hydroxyl group off of one of the glucose here, and the hydrogen off of the hydroxyl group on the fructose. That makes our sugar, and then makes our water, and then we've got an open bond there. We can join them together. In the case of carbohydrates, that linkage is called a glycosidic linkage. Anytime you see the term, the prefix glyco, that usually refers to glucose or some kind of sugar, just so you know that. Polysaccharides have a couple of different roles depending on what kind they are. Polysaccharides can be storage forms of glucoses, like you see here in plant starch, okay, or in glycogen, which is what animals store in their muscles for stored glucose. Notice that you've got all of these different glucoses that are joined together. You see there's an oxygen there. That's the oxygen that's in the ring, and here's the oxygen joining those together. This is called an alpha linkage, okay? And so notice that they're all exactly in the same order or in the same direction, all right? And this is going to be a fuel molecule for animals and plants, or animals and plants. But then you have other molecules like cellulose that's shown here that are used for structure. The cellulose is what makes up the cell walls of plants, and it's found in lots of other things as well. And cellulose molecules, notice that here you have the oxygens are arranged a little bit differently than you see up here, and it looks like every other one is going in the opposite direction of the previous one. That's called a beta linkage, and beta linkages lend themselves to cross linkages or hydrogen bonding between the strands there, and that makes a really strong fiber that can be used as a structural support. And so plant cell walls, there's another common polysaccharide that's used for structure called chitin that is found in arthropod exoskeletons and in fungi cell walls. That's also somewhat, it's a little bit different than this one. It's obviously it's a different molecule, but it's also a storage molecule rather than one that's used for fuel. Lipids are another category. Again, they're relatively simple in terms of their construction, okay? But the big thing to remember that even though they're biomolecules or organic molecules, they do not form true polymers. Some of them can be pretty big, but they're not polymers, and true polymers in the sense that that carbohydrates can be, and proteins and other molecules, okay? They're generally nonpolar and hydrophobic. Okay, they have, like it says here, their unifying features having little or no affinity for water. The reason is they're mostly hydrocarbons, okay, and hydrocarbons are nonpolar. The most biologically important ones are fats, phospholipids, steroids. There are others as well, like waxes and things like that. We're going to focus mostly on the triglycerides and the phospholipids, and then on the steroids, which are found in hormones and things like that. Fats are constructed of two types of smaller molecules, glycerol and fatty acids. Glycerol is actually a sugar or an alcohol that has 3 hydroxyl groups. You can see here in the molecule, in the structural molecule there. And then fatty acids are long hydrocarbon chains with a carboxyl group on the end. That's how they come there in acid. Do you see any oxygen anywhere except in that carboxyl group in the fatty acid? No, you don't. Okay, so that's the only place they'll have oxygen. So again, lots of carbon, lots of hydrogen, very, very little oxygen that you'll find in fats. When that bond is formed, again, by dehydration synthesis, that forms what we call an ester linkage between the glycerol and the fatty acid there. And this one right here is a fat molecule. So you may have heard of triglycerides. That's a typical fat molecule that you would find in animals and plants and lots of different food. If you look at the nutrition facts, you'll look at the fats basically, and this is a fat that you would see in there. Okay, there are a number of different fatty acids. Some fatty acids are longer than others. Some of them are what we call saturated fatty acids, like this one here, that have all single bonds between carbons and lots of hydrogen attached. Saturated means that they are saturated with hydrogen. They've got hydrogen in every spot that there's a place that you can put a hydrogen. And they're solids at room temperature, so things like butter. Animal fat generally is saturated fat. Unsaturated fatty acids have one or more double bonds in the chain. That causes them to bend, okay? And these are called unsaturated. They are liquids at room temperature. Big difference is when they get cold, these straight chain fatty acid tails can cluster really close together, pack in really, really tightly. But the ones that are bent, the unsaturated ones that are bent, prevent them from packing as tightly, so they can't get into that solid form very easily. And so these are generally liquids at room temperature. Okay, unsaturated fatty acids are better for you in terms of your health than saturated fats are. Saturated fats are associated with things like atherosclerosis, hardening of the arteries, high blood pressure, heart disease, that kind of stuff. Okay, phospholipids are somewhat like triglycerides, but they only have 2 fatty acids instead of 3, and in that third spot on the glycerol, they have a phosphate group attached. Okay, phospholipids are special molecules that are found in cell membranes. They are what we call amphipathic. Let me write that on here for you, if I can find a place to write. Amphipathic. Amphipathic. And what that means is they've got a hydrophilic portion and a hydrophobic portion. And so the hydrophilic portion in the cell membrane is going to be on the outside and inside of the cell membrane, because that's going to be the watery substance. And then the nonpolar hydrophobic tails are in between. And that forms a nice barrier with a liquid substrate that you're involved in. The last category of lipids is steroids. Steroids have a carbon skeleton that's mostly carbon and hydrogen. It'll have some other groups attached to it, but they're basically 4 fused rings. The one that's shown here is cholesterol. It is an important component of animal cell membranes. Methyl group in that is definitely nonpolar, and so this is going to attach to the hydrophilic part of the phospholipids, and this part will be in between the hydrophobic tails of the phospholipids. It's really important to maintain the stability of the cell membrane, and we'll talk more about how that works when we talk about cell membranes. Cholesterol is essential in animals. It's found in all animal cell membranes. But if you have too much of it, if it's in your blood, then it can contribute to atherosclerosis like the triglycerides can as well. So we're going to stop there. That takes care of our carbohydrates and lipids. And the next video we'll talk about proteins and nucleic acids.